failed to upload or whatever. So I need to redo it. And it's just going to be over the eye. Okay, so the last two topics of this quarter are going to be on the special senses, which are actually taste, smell, sight, which is vision, and also equilibrium. And so, and then we're going to talk about the endocrine system. And the endocrine system that plays in nicely because, especially with the nervous system, because the endocrine system interacts with the nervous system to actually kind of allow long-term regulation of different aspects of the body. So you'll remember that sensory receptors are actually modified neurons, right? So these are modified neurons. And we talked about some examples of these. So we talked about proprioception in the muscles that allow our body to know whether the muscles are at the appropriate tension or if they're overstretched or understretched to keep muscle tone. And then we talked about touch receptors and pain receptors on the surface of the body that send signals to the somatosensory cortex. So we are now going to focus on special senses. And these are sensory receptors that have an organ associated with them. So for example, taste, which would be the tongue, right? Smell, which would be the nose. Vision, which would be the eye. And also equilibrium. So we're going to talk about, oh, and hearing. So we're going to talk about the ear and the inner ear and how it is both responsible for the ability to see or to hear sounds, but also it gives us a sense of our relationship to gravity and whether or not we are moving or if we are still. So equilibrium is really important, actually. People that have vertigo, for example, can be very, very debilitating to them. Okay, so sensory receptors, modified neurons, and we're going to talk specifically when we go to each of the special senses, we'll talk about the type of receptors and then the transduction of that stimulus energy into an action potential. Okay. So we're going to start with the eye today. And when we look at the structure of the eye, we have accessory organs or accessory structures. So for example, we have the eyebrow. And the eyebrow is really interesting because if you've ever worked in construction or anything else that's really dusty, or even like you're outside digging, you'll know that your eyebrows actually collect debris. And so they actually help prevent getting dust and stuff into your eyes. We also have the eyelashes, which are actually very sensitive to touch. And so they actually not only keep stuff from getting into our eyes, but they also allow us to blink. So if anything gets near the eyes and they touch your eyelashes, you'll blink as a protective mechanism. And the eyelashes have actually a special name, and this is called the palpebrae. I practiced this. Palpebrae, so palpebrae, or palpebral, that's the eyelashes. Okay, we also have some glands. So the big one that we think about when we make tears are the lacrimal glands. And these glands have ducts that release tears. And tears actually have multiple functions. One, they keep the eye from drying out. Two, they also have an immune function. So, for example, we have lysozyme. That's a this is a Z, lysozyme, and enzymes. And these break down bacteria. We also have antibodies in the tears, and we'll talk about how specialized antibodies can bind to specific microorganisms and allow us to break them down, and so that kind of prevents them from the eyes from getting infected. If we look at the structures that we've been talking about in your book, right? So this is the eyelid, the eyelashes and the eyelid, okay? And so where the two eyelids come together is the palpebral fissure, where the two eyelids come together. And then we can talk about the lacrimal caruncle. So the lacrimal caruncle is this sebaceous gland, and it also has sweat glands too. And so this is where you get sand in your eye or sleep in your eye, right? So this is actually, this tissue right here in the corner of your eye is actually a gland. And the reason why, sometimes when you wake up, is it's not only just dry tears, but you can actually get sebaceous secretions drying up. And so like when you wake up, you have to clean that area out. And sometimes if the tears do not drain away, which we'll talk about, they're draining out through a hole here in the side of the eye, then sometimes you can get, like babies, for example, they wake up and they have totally gunky eyes. And so that can be due to that, those secretions not leaving the eye and then they get dried out. Okay. We also have a structure which you can see here, which they talk about, which is the conjunctiva. And so the conjunctiva is a mucous membrane that covers the eye except for the cornea. We'll talk about what the cornea is in a minute. And it also lines the area under the eyelashes, so under the eyelids. Okay. So it actually covers the whites of the eye right here. And it is, sometimes it can swell up. And so sometimes when you have allergies, for example, this will become bright red and it'll become swollen, for example, it can be really painful. You can also get viral or bacterial infections in the conjunctiva. And what is that called? If you get a viral or bacterial infection, it's called pink eye, right? Or conjunctivitis, right? So conjunctivitis is pink eye. And it's important to realize that it can be both viral and or bacterial. And the bad thing about pink eye is that it is highly contagious. And so if you have a kid in school, right, they send them home, right? And oftentimes they'll make them go to the doctor to get some antibiotics. But oftentimes the pink eye is actually not bacterial and it's viral in nature. And so I think that the doctors are trying to get people to stop just kind of using too much antibiotics in the eye, just because a lot of it is viral and the antibiotics didn't do any good. And the bacteria might become more resistant to the antibiotics over time. So the schools kind of push you to get antibiotics because then they'll let your kid come back to school. But it doesn't matter because it could be viral and then that could be contagious. So 7 days is like the amount of time for it to kind of resolve itself by itself. Okay. So another image here of the eye, kind of a side view. So here we see, this is the conjunctiva. And notice that it actually comes and folds under the eye here and under the eyelid here. And so that would be actually where, that fold is actually where contact lenses would sit in the eye. And that folding prevents friction between the eyelid and the eye, right? Unfortunately, there are some illnesses where you can actually get bacteria up underneath the eyelid, and you can get a bacterial infection, which is called trachoma. So trachoma is a bacterial infection underneath the eyelid. And it is actually a type of bacteria which is called chlamydia. And chlamydia is very highly contagious. This would be highly contagious as well. And in countries where they don't have a lot of hygiene or a lot of medical assistance, this becomes really a problem because worldwide, 1.9 million people go blind because of this disease, or are blind go blind because of this disease. And the reason is because it causes irritation underneath the eyelid, and it can cause the eyelid to actually turn under. And so you actually have the eyelashes scratching the cornea, and that scratching of the cornea leads to blindness like this. So this is not in your book, obviously, but so this would be an infected membrane that is causing the eyelashes to actually scratch the cornea right here, for example. And that scratched cornea over time will just cloud up, and people can get, and I can think of no other worse thing to have happen, because you can imagine how painful that must be, because we have these pain receptors on our cornea. And so that this would just be a really horrible disease to have. And so here they say that it's transmitted via fleas in a household, from, and then also from children to parents. But the good news is this is treatable with antibiotics. So only about 1,000 cases a year happen in the United States. Okay, so this diagram shows the lacrimal gland. So would the lacrimal gland be an example of an exocrine gland or an endocrine gland? Exocrine, right? So you notice there that it has excretory ducts. And so these ducts are what create the tears. And then they have to drain here, and the draining is via the punctum. So think of like a puncture, right? So think about a little hole here in the edge of your eye, and that is where the tears drain. So you need to know the lacrimal punctum. This is where tears drain into the lacrimal sac. And then they go into the nostrils. So you see here, they can go down, and so if you cry a lot, for example, you'll notice that your nose will start to run, right? So if you're producing a lot of tears and these tears are draining, then it starts to come out your nose, and that's because it drains into the nose. So it drains via the nasolacrimal duct. Okay. So we talked about skeletal muscles, and so we do have some extrinsic eye muscles, and these are skeletal muscles. So we can talk about the extrinsic eye muscles. And these are skeletal, and there are 6 of them. Extrinsic means that they're on the outside. We also are going to talk about intrinsic eye muscles, which are on the inside. So for example, the iris and the ciliary body, which we've kind of talked about in terms of autonomic reflexes, are intrinsic, but these are extrinsic. And so they attach onto the eyeball and cause the movement of the eyeball. So a lot of these have the term rectus in them. So what do you think rectus means? Do you remember? Straight, right? So you'll notice the striations, right? They run in a straight line. So for example, the rectus, the lateral rectus is going to be able to let you move your eyes to the side, and our eyes kind of move together, and that's important so that we can focus our vision. If your eyes move independently of one another, then you can't see very well, and you get what is called double vision. And so there is another type of muscle here. There's two obliques, and the inferior and the superior obliques. And oblique also refers to the arrangement of the muscle fibers, right? So it's at an angle. And so these oblique muscles are kind of interesting because they do the exact opposite of what you would think. So for example, the inferior oblique, you think low, right? So you think you're going to look down, but it actually, the way that it moves the eye is it actually pivots the eye up. And so the superior oblique is also really interesting because it crosses through, its tendon crosses through this structure right here, which we'll look at in lab, which is called the trochlea. And it's like a pulley. So trochlea means pulley. And we talked about the trochlear notch in the elbow. So we have another trochlea in the eye. And it also does the opposite. So instead of actually causing you to look up, it actually causes you to look down and a little bit medial. Lateral, look down and off to the side. And so those eye muscles have to be coordinated with the eyes together so that you can see the correct image. Now there is a problem, which is called strabisma or strabismus, and this is being cross-eyed. Oops. And sometimes, for example, babies are born cross-eyed. And what that means is that one of the muscles is not working properly. It could also be that one of the muscles is weak. And so what happens is that the eyes turn in, right? Because this lateral muscle is weak and not able to stabilize the eye and keep it from turning in. And so oftentimes they have, exercises that you can do with your eyes to strengthen these eye muscles. But oftentimes it causes, you have to have surgery, right? So you have to go in and surgically kind of fix the eye so that the eyes can focus forward. Okay. So we're going to talk about the structure of the eyeball itself. And in the eye, we have 3 tunics. These are also layers. So the first tunic is called the sclera, and it is the outer layer. So it is dense connective tissue. It is what the extrinsic eye muscles attach to. And where light has to enter the eye, the sclera is transparent, and it's called the cornea. So the cornea is actually part of the sclera, but it is the transparent part of the sclera. Now, dense connective tissue does not have any vascular supply to it, and so the cornea is actually easily transplantable from one person to the next because the immune system cannot get in there to reject the organ, right? So it's no blood supply, and it's easily transplantable. So if it gets scratched, transplantable. I don't know if that's an ibble or an ebble. Transplantable. So if it does get scratched, those people that are blind, perhaps they could get a cornea transplant to fix their blindness. Okay. The other thing is that there are pain receptors here. And this is really important because it makes us protect our eye. So if you have ever had a scratched cornea, you know how painful it is. like it burns and it stings and it'll keep you up at night and you can't sleep because of the pain, right? So it's very, very bad to scratch the cornea. And oftentimes when you scratch the cornea, they're also worried about infection. And they're also worried specifically about the infection that can lead to what disease? If you get a scratched eye, they want to know what vaccine are you up on? Anybody know? Go in and they say, are you up on your tetanus vaccine? Yes. So tetanus can be gotten off of plants and stuff if it scratches your eye, so you can get tetanus from that, and that's different than tetanus muscle contraction. But it does cause lockjaw, but it is a bacterial infection, and the bacteria specifically produce toxins that affect skeletal muscles. Okay. So the sclera is actually where the extrinsic muscles attach, because it's the outer part. The next part is called the choroid, and the choroid is the middle layer, and it is dark in color, and it is highly vascular. And it is actually the reason when you get your picture taken and the flash hits just right, that your eyes look red, right? Because it is where the blood is. So the choroid provides nutrients and oxygen to the sclera, because remember the sclera, the dense connective tissue, has living cells in it, right? So they actually have to get nutrients from the choroid. Now, the part of the choroid that makes up the iris is colored, right? So the iris is smooth muscle, and it is the colored part of the eye. Eye. So melanin pigment gets deposited in the eye, right? In the iris. And this controls the diameter of the pupil. So the pupil is just the opening where light enters the eye. Right? And there's actually two different muscles. And remember, they're innervated by the autonomic nervous system, and they can cause dilation or constriction depending upon which division of the ANS is stimulating the eyes. Okay, another part of the choroid is called the ciliary body. And the ciliary body actually has tendons that connect. It is also smooth muscle, and it connects to the lens. So smooth muscle with tendons that connect to the lens. So this allows for accommodation. So that is the ability to see close up and far away. Okay, so it actually changes the shape of the lens depending upon how it is contracting or relaxing. Now the ciliary body has another function. There's actually capillaries here, because remember it's highly vascular. So there's capillaries that produce the fluid, or part of the fluid in your eye, called the aqueous humor. And we'll talk about the aqueous humor in a little bit. But this is the watery fluid that is in your eye, and it's like right underneath or it's like surrounding the, it's right underneath the cornea, but also surrounds the lens of the eye, and that's the aqueous humor, versus we'll talk about the vitreous humor in a little bit. Okay, and so then finally, the innermost layer is called the retina. And it consists of nervous tissue. So we'll talk about the different types of receptors in the eye, as well as the neurons that connect those receptors to the optic nerve. Okay, so this is very thin. And it gets the nutrients from the choroid. So there is a problem if the retina detaches. That means that it is coming free from the choroid, and that means that those neurons are not going to get any nutrients, and the person is going to go blind. So when you get retinal detachment, that means that the neurons are going to die. And so they do have this great new treatment where they actually go in and with a laser, they can reattach the retina if they catch it before the neurons start to die in abundance, so that you can regain your vision from retinal detachment now. Okay, so this is the diagram, okay? So this shows the 3 layers, right? The sclera, the choroid is the dark red, retina, nervous tissue is always yellow, right, in the book. So whenever they draw nervous tissue, it is always yellow. When we look at it, it's kind of more pink, a little bit pinkish when we look at the dissection of it. So another structure that we need to talk about is the lens. So the lens has an outer epithelial layer. So there are living cells on the outside of the lens, but consists of the inside, it consists of protein. So the inside is not cellular, but consists of crystalline protein. Okay. So this protein called crystalline, right, has a particular shape that allows light to be transmitted through it. And if you have, or as you get older, or if you have genetic diseases, the crystalline proteins can clump. And when they clump, they cause a clouding of the lens. And what is that called? Cataracts, yes. So the clumping of the proteins causes cataracts. Now you can get cataracts also from too much sun, right? So that might be the reason why older people tend to get cataracts, is just the, how much UV radiation. So they always recommend that you wear sunglasses with UV radiation protection, because that will keep you from getting cataracts when you get older, right? But as you can also have juvenile cataracts, so you can have it happening in children, and that seems to be a genetic propensity towards cataracts. Okay, so the lens is located right here, right? And you notice that we have these little tendons that connect the ciliary body to the lens, and then this is the iris. This is the colored part of the eye, and then the pupil is where light enters the eye, and then it has to be focused. The light has to be focused as it comes in on the back of the eye and on the retina. So that the lens is what focuses it by changing shape. So if the lens changes shape, it will focus the retina. Sometimes the lens is not able to change shape, or the eyeball is the wrong shape. And then the image gets projected too far back or too far close, right? And that's the difference between near vision and far vision. Okay, so these are the smooth muscle, and you can actually see the way that they would work by looking at their fascicle arrangements. So you don't need to know the difference between these two, but I think it's kind of interesting. So these are the circular ones. So when they contract, they are like a sphincter. And so the sphincter causes a closing of the pupil. And then if you look at the arrangement of the fibers on this one, you'll notice that when it contracts, it's actually going to open up and pull and allow the pupil to get larger. So remember, the parasympathetic and the sympathetic have antagonistic effects on the iris. And the iris is an example of an intrinsic muscle. So I'll put iris and the ciliary body are intrinsic muscles of the eye. As opposed to extrinsic muscles. Okay. Okay, oops. So we're going to talk about the retina now, and the retina consists of 3 different types of neurons. We have first the photoreceptors. Oops, my pen's not working very well today. Let's erase it. Okay, so we'll talk about the photoreceptors in more detail, but there's 2 different types of photoreceptors. There's the rods and there's the cones. Now the photoreceptors then connect to neurons that are called bipolar neurons. And we talked about bipolar neurons when we were talking about the different types of neurons. So this means it's got like 2 axons coming out from a central cell body, right? And then we have what are called the ganglion neurons. So these specifically connect to the optic nerve. So they're actually the axons. So these contain the axons. The optic nerve contains the axons of these neurons. Okay. So the weird thing about our eyes is that they are arranged not how you would probably arrange it if you were building an eye to detect light. Because what happens is when we look at our say this is our photoreceptor, and then we look at the bipolar cell, right, and then another neuron, right? So this would be my photoreceptor, this would be my bipolar, and this would be my ganglion. Light actually comes in this direction. So for some reason, the photoreceptors are not out front. Like if you were going to design an eye, you put the photoreceptors out front and you put all the wires to the back. And that actually is the way that the eye works in the octopus and the squid and other cephalopods, is you have the photoreceptors out front and all the neurons taking the action potential into the brain coming out the back. But our system is designed differently. So all this nervous tissue has to be very transparent, and light actually has to travel through the bipolar neurons and the ganglion neurons before it actually gets to the photoreceptor. Okay. So if we look at a diagram of this in your book, this is what it looks like. Okay. So notice that the photoreceptors are on the back side of the retina. Light would come in this direction. This is my bipolar cells. These are my ganglion cells. These are the rods and the cones. And the rods and the cones, you'll notice, have different shapes. So rod-like is rectangular, and then cone-like, right, would be like a cone, right? So that they're named after their shape. So we do have part of our retina that does not contain any photoreceptors, and this is what is called the optic disc. So the optic disc is where the optic nerve leaves the eye, and there are no photoreceptors in this area. So this is what we call the blind spot. So there truly is a blind spot. So like when you're driving, right, and somebody is in your blind spot, it's really annoying because you keep having to look back to make sure they're there, right? So when you're driving, you try to stay out of people's blind spots, right? And so you want to speed up rather than hanging right here. Because literally, when a person looks ahead and what they see in their peripheral vision is what they think they should see, and the eye will actually fill in like an empty street, right? And so we'll do a little experiment in lab on Friday that looks at that, where that is and how you can detect where your optic disc is located. Okay. So again, this is actually looking at not a diagram, but this is actually a microscopic view of the retina. So you can see what it would look like underneath a microscope. Okay. And oftentimes you go into the eye doctor and they take a picture like this. And so what they are looking for is to make sure that you do not have any constrictions in the blood vessels of your eye. And they're also kind of looking for to see if the retina looks healthy, because this is the type of picture that they can use to say, you have retinal detachment and we need to go in and repair that. So you might see a lighter colored area or a darker colored area. The macula lutea, I believe, is where you have a higher concentration of photoreceptors located right here. That's more in the center of the eye rather than on the peripheral. So this is my optic disc. So this is actually where the blood vessels come in, right, and help supply nutrients to the retina. Okay, so let's talk about the chambers. So we have what is called the anterior chamber. And obviously this is near the front of the eye. And it contains aqueous humor. Okay, this is produced from blood in the capillaries of the ciliary bodies, right? We broke that down. So this produced from blood, and it is circulated and reabsorbed. So this can contribute to what is called intraocular eye pressure. So when you go to the eye doctor, they take that little picture of the blood supply to the retina, but they also do what to check the pressure in your eye? Right, so they put you up at the machine and they go poof, right, and you blink, right? And what they're doing to see is if the cornea is going to depress a little bit. If it depresses too much or too little, and specifically if it depresses too little, then that means you have too much pressure in your eye. So that is a disease which is called glaucoma. And it can damage the retina and cause blindness. Okay. So specifically people with diabetes tend to have a problem with glaucoma. So they have to take drugs in order to keep that amount of aqueous humor in their eye normal, because it's just not being recirculated and reabsorbed fast enough, and too much is being produced. And so it causes an increase in eye pressure. Okay, so in the posterior chamber, so that's the back of the eye, we have what is called the vitreous humor. So vitreous means thick, viscous, and so this is gel-like. And so when we do the cow eye dissection, you're going to notice that all this like gel-like stuff comes out of the eyeball, right? And this actually gives the eyeball its shape, right? So it has to be filled with something so that it provides, its shape, its pressure. And then this also is where, like the aqueous, we actually have light being transmitted through this gel-like stuff as it is being transmitted onto the retina. So it has to go through the vitreous humor. And then this is produced embryonically. And it is not circulated. So it is produced as when you're an embryo, and then you have it for the rest of your life. So if we look at these chambers of the eye, the posterior chamber is obviously bigger. This is the anterior chamber. And so they note here how it is being produced, and then it comes out through the iris, and then it gets reabsorbed and put back into circulation there. So underneath the cornea and kind of in this area right here, that would be filled with the aqueous humor, and the vitreous humor is what is in the posterior segment. So the most of the stuff is going to be vitreous humor that's going to come out of the eye. Okay, we already talked about the lens, so we won't talk about that. This is cataracts, clouding of the lens. Okay, so let's talk more about the photoreceptors. So the photoreceptors are modified neurons. And they have inside of them special organelles that have associated with them photopigments. So we have pigments that are able to absorb light energy. So this is kind of like plants. Plants have chloroplasts that have chlorophyll, and they absorb light energy to produce sugars, but we absorb light energy in order to produce an action potential so that we can see images. And so the pigment is actually called rhodopsin. And rhodopsin can be converted in the light or of the dark into what is called retinol and opsin. So opsin is a protein, and it varies between the rods and the cones, and it also varies between the different types of cones. So we have red and green and blue cones. Okay, so that's what varies. The retinol is essentially what type of vitamin? What vitamin is retina or retinol? Sometimes you see it in skin creams.
Is it like keratin?
Yes, it is. Vitamin. Nope. Nope.
Vitamin K?
Nope. Vitamin A. Okay. So yes, we can get vitamin A, right, from eating vegetables that are orange. So like it is true, carrots, we need orange vegetables like carrots or squash or something to get our beta carotene so that then we can produce retinol. And so truly there are people that are so malnourished in this world that if they do not have vitamin A in their diet, right, they will go blind. And so there's actually been, in India, for example, okay, that's all I hear. Okay, vitamin A, they actually decided they were going to genetically modify rice and produce what is called golden rice. And golden rice has vitamin A in it. So the idea is that you could actually give people vitamin A in golden rice to keep them from going blind. So yes, you can go blind if you don't have that nutrient in your body. Okay. So notice that this chemical reaction goes both ways. Okay, so this is light. So when rhodopsin absorbs light, retinol and opsin dissociate. They come apart. So it uses that light energy to change the shape, and it's the shape of the retinol so that it will come apart from the opsin. And in the dark, it goes the opposite way. So in the light, this is sometimes referred to as the bleaching reaction. So you can say that the rhodopsin gets bleached as it turns into retinol and opsin. So this is what happens, for example, when you go outside and you're in the bright sunlight, and then during the summer and you walk inside and you can't see anything, and you turn on the light because you think it's dark in there, right? And you still can't see anything. That's because your retinol and opsin have all disassociated, and they've all become, that pigments become bleached. And so you have to stand there for a few minutes in order for this chemical reaction to go the other way. So there's an enzyme present that puts those two things back together again. Right? So that's the bleaching reaction. Okay, if we look at the type of receptors, we have the majority of our receptors are rods, right? And the rods are responsible for black and white vision. And they have only one type of opsin. Right? And the other thing is that these are very sensitive to low levels of light. Right? So this is what happens when the sun goes down and it becomes dusk, right? All the colors just bleach away. And at night, you're kind of left with the rods. And so you do not see colors very sharply at night. And so this is responsible for night vision. The other thing is that the rods are more concentrated in the periphery of the retina. So if you take an astronomy class, they always teach you, if you see something really dim, you don't look straight at it. You actually look at it from the side, because you'll be able to see a dim star in your peripheral vision, and then when you turn to look at it, it disappears. And that's because in the center of our retina, we have cones that are not able to fire under low levels of light. So the rods have an important function, and so cats and dogs, for example, only have rods, and so that's why they cannot see color. They can't see color, okay? But they're really good at seeing in low levels of light. Okay, so we have the cones, and we have red, green, and blue. And for the life of me, I cannot figure out why these aren't the primary colors. Doesn't make sense to me. Why is it blue? I don't know. But anyway, red, green, and blue are the different types of cones, and they have different opsin, and they absorb different wavelengths of light. So the overlap allows us to see different colors. The way that the red cones would fire, the blue cones would fire, and we would see, what do red and green make? Purple? No, red and blue make purple. What? I don't know. I think green. Anyway, so the different types of cones that fire will give you the difference. Now, some people are lacking one or the other. So for example, they could only have green cones, right? And so if they only had green cones, they're not going to be able to see the difference between red and green because of the overlap. Okay. So these are obviously color vision. They're less sensitive to light. They're at the center of the retina. Right? And this is during the daylight vision. So birds, for example, are active during the day, and they also have bright coloration on them to communicate with one another, and so they actually have color vision. And they can also see like red flowers, right? They're attracted to them. And insects also have color vision because that's how they're attracted to the different types of flowers, right? Color vision. Okay, so when we look at photoreceptors, this is the difference between the rods and the cones, right? Their location. And these little discs are actually what actually take or have the visual pigments in them. And those discs look really, really, really similar to the chloroplasts in plants that contain chlorophyll. So they're just discs with pigments. Oops. Okay. So this is a showing the discs that would be found in a rod. And then they show, so these are organelles and they're bound by membranes. And so this is actually a plasma protein that sits, plasma membrane protein that sits in here. So this is the opsin and this is the retinol. And so they sit there until light hits them. And then when the light hits them, that's when we get transduction. Okay. So here you can see that there's actually a change in the structure of the retinol. So this would be where it is attached, right? And it looks like this. You don't need to know that, but the retinol would look like this. But when it absorbs light energy, it looks like that. And that causes it to come apart. Okay, so transduction in photoreceptors is very strange. So we're going to talk about transduction now. Okay, so this is very different from all other receptors in the body. So the first thing that makes it different is that the photoreceptors are depolarized in the dark. So they're actually depolarized when there is no light. Most receptors depolarize when they absorb the energy. Like, so our smell and taste would be that way, touch would be that way. They're not depolarized when they're doing nothing. But the photoreceptors are depolarized when there is no light, okay? So what that means is that they are also releasing neurotransmitters into the synapse. And into the synapse with the bipolar cells. Okay. So this neurotransmitter must be inhibitory because we can't have action potentials being sent to the brain when there is no stimulus. So it is an inhibitory neurotransmitter, so it prevents action potentials from being sent. Okay. So when stimulated, okay, the photoreceptors become hyperpolarized. So I'll just put become hyperpolarized, or they become hyperpolarized. No neurotransmitter is released. Okay, and an action potential is produced. So I'll just put AP, action potential is produced. So they actually become hyperpolarized when stimulated rather than depolarized. They stop producing neurotransmitters, and that allows an action potential to be produced. And I don't know if anybody knows why photoreceptors do the exactly opposite of all other receptors in our body. Okay, so if we look at a diagram of this, so this is my photoreceptor in the dark. And so notice sodium is flooding in. And sometimes they call that flooding in of sodium the dark, what do they call it? The dark current. So there's sodium floods in when it is, when there's no light, and then it releases a neurotransmitter, right? And then it prevents the bipolar cell from sending neurotransmitters, excitatory neurotransmitters, to the ganglion cells. Okay, so that's in the dark. And then in the light, what happens? The sodium channels stop, or they close, right? And then it becomes hyperpolarized. No neurotransmitter is released. And then notice how in the bipolar cells, now there's neurotransmitters being released, and then an action potential is set into the central nervous system via the optic nerve. Okay, so to summarize this, I'm just going to show you a short little 10-minute video. That's what we'll do for the rest of the class. And this is from that A&P crash course on Khan Academy, which talks specifically about vision. Okay, so he's going to ask you to stare at this screen for a few moments.
Illusions use patterns of light or perspective to exploit the disconnect that exists between sensation and perception, between what your eyes see and what your brain understands. But not all illusions work that way. Some produce ghost effects or after images that take advantage of glitches in the physiology of human vision, like this flag. I'm not trying to make a political statement here, and I'm not going to ask you to swear allegiance to the Republic of Hank or anything. I mean, if I was going to start my own country, my flag would be way cooler than that. Not that I've thought about that a lot. And now look at this white screen. If you looked at that flag for at least 30 seconds without moving your eyes, you'll see something, even though the screen is blank, an after image of the flag. But instead of being turquoise and black and yellow, it's red, white, and blue. Okay, so that's pretty cool, but I'm not here just to entertain you. This kind of illusion is actually a great way to explain your very complex sense of vision. And I do mean complex. Nearly 70% of all the sensory receptors in your whole body are in the eyes. Not only that, but in order for you to see, perceive, and recognize something, whether it's a flag or a handsome guy in glasses and a sport coat sitting behind a desk, nearly half of your entire cerebral cortex has to get involved. Vision is considered the dominant sense of humans, and while we can get along without it, and it can be tricked, what you are about to learn is not an illusion. When we talked about your sense of hearing, we began with the mechanics of sound. So before we get to how your eyeballs work, it makes sense to talk about what they're actually seeing. Light bouncing off of stuff. Light is electromagnetic radiation traveling in waves. Remember how the pitch and loudness of a sound is determined by the frequency and amplitude of its waves? Well, it's kind of similar with light, except that the frequency of a light wave determines its hue, while the amplitude relates to its brightness. We register short waves at high frequencies as bluish colors, while long, low frequencies look reddish to us. Meanwhile, that red might appear dull and muted if the wave is moving at a lower amplitude, but super bright if the wave has greater amplitude and thus higher intensity. But the visible light we're able to see is only a tiny chunk of the full electromagnetic spectrum, which ranges from short gamma and X-rays all the way to long radio waves. Just as the ear's mechanoreceptors or the tongue's chemoreceptors convert sounds and chemicals into action potentials, so too do your eyes' photoreceptors convert light energy into nerve impulses that the brain can understand. To figure out how all this works, let's start with understanding some eye anatomy. Some of the first things you'll notice around your average pair of eyes are all the outer accessories, like the eyebrows that help keep the sweat away if you figure out your headband or racquetball, and the super sensitive eyelashes that trigger reflexive blinking, like if you're on a sandy beach in a windstorm. These features, along with the eyelids and tear-producing lacrimal apparatus, are there to help protect your fragile eyeballs. The eyeball itself is irregularly spherical with an adult diameter of about 2.5 centimeters. It's essentially hollow, full of fluids that help it keep its its shape, and you can really only see about the anterior sixth of the whole ball. The rest of it is tucked into a pocket of protective fat, tethered down by 6 strap-like extrinsic eye muscles and jammed into the bony orbit of your skull. Well, all this gear generally does a fantastic job of keeping your eyeballs inside of your head, which is good. On very rare occasions, perhaps after head trauma, or even a really intense sneeze, those suckers can pop right out, a condition called global luxation, which you really do not want to Google. Just sit here while you Google it. Now, you don't need to pop your eyeball out in order to understand how it's structured. I'll save you the trouble and tell you that its wall is made up of 3 distinct layers, the fibrous, vascular, and inner layers. The outermost fibrous layer is made up of connective tissue. Most of it is that white stuff called the sclera, while the most anterior part is the transparent cornea. The cornea is like the window that lets light into the eye, and if you've ever experienced the excruciating pain of a scratched one, you know how terrible it can be to damage something so loaded with pain receptors. Going down a little deeper, the wall's middle vascular layer contains the posterior choroid, a membrane that supplies all of the layers with blood. In the anterior, there's also the ciliary body, a ring of muscle tissue that surrounds the lens, but the most famous part of this middle layer is the iris. The iris is that distinctive colored part of the eye that is uniquely yours. It's made up of smooth muscle tissue shaped like a flattened donut and sandwiched between the cornea and the lens. Those circular sphincter muscles, yeah, that's right, you got sphincters everywhere, contract and expand, changing the size of the dark dot of your pupil. The pupil itself is just the opening in the iris that allows light to travel into the eye. You can see how an iris protects the eye from taking too much light, and if you shine a flashlight in your friend's eye in a dark room, their pupils will go from dilated to pinpoints in a couple of seconds. Light comes in through the cornea and pupil and hits the lens, the convex transparent disc that focuses that light and projects it onto the retina, which makes up the inner layer of the back of the eyeball. Your retinas are loaded with millions of photoreceptors, which do the crucial work of converting light energy into the electrical signals that your brain will receive. These receptor cells come in 2 flavors of rods and cones, which I'll come back to in a minute. But the retina itself has 2 layers, the outer pigmented layer that helps absorb light so it doesn't scatter around the eyeball, and the inner neural layer. And this layer, as the name indicates, contains neurons. Not only the photoreceptors, but also bipolar neurons and ganglion neurons. These 2 kinds of nerve cells combine to produce a sort of pathway for light, or at least data about light. Bipolar neurons have synapses at both ends, forming a kind of bridge. On one end, it synapses with a photoreceptor, at the other, it synapses with a ganglionic neuron, which goes on to form the optic nerve. So say you've just been hit with a blinding flashlight beam. That light hits your posterior retina and spreads from the photoreceptors to the bipolar cells just beneath them to the innermost ganglion cells, where they then generate action potentials. The axons of all those ganglion cells weave together to create the thick, ropey optic nerve, your second cranial nerve, which leaves the back of your eyeball and carries those impulses up to the thalamus and then on to the brain's visual cortex. So that's the basic anatomy and event sequencing of human vision. But what I really want to talk about are those 2 types of photoreceptors, your rods and your cones. Cones sit near the retina's center and they detect fine detail and color. They can be divided into red, green, and blue sensitive types based on how they respond to different types of light. But they're not very sensitive, and they really only hit their activation thresholds in bright conditions. Rods, on the other hand, are more numerous and more light sensitive, but they can't pick up real color. Instead, they only register a grayscale of black and white. They hang out around the edges of your retinas and rule your peripheral vision. Since these receptors function so differently, you might not be surprised to learn that your rods and your cones are also wired to your retinas in different ways. As many as 100 different rods may connect to a single ganglion cell, but because they all send their information to the ganglion at once, the brain can't tell which individual rods have been activated. That's why they're not very good at providing detailed images. All they can really do is give you information about objects' general shape, or whether it's light or dark. Each cone, by contrast, gets its own personal ganglion cell to hook up with, which allows for very detailed color vision, at least if conditions are bright enough. And all this brings us back to that weird flag. Why does staring at this flag and looking at an empty white space make us see a phantom flag of different colors? Well, it begins with the fact that our photoreceptors can make us see after images. Some stimuli, like really brilliant colors or really bright lights, are so strong that our photoreceptors will continue firing action potentials even after we close our eyes or look away. The other part of the illusion has to do with another bug in our visual programming. And it's just that our cones can get tired. If you stare long enough at a brightly colored image, your cones will receive the same stimulus for too long. Basically stop responding. In the case of the flag, you looked at an image with bright turquoise stripes. Because your retinas contain green, red, and blue sensitive cones, the blue and green ones got tired after a while, leaving only the red cones left to fire. Then you looked at a white screen. That white light included all of the colors and wavelengths of visible light, so your eyes were still receiving red, green, and blue light, but only the red cones were able to respond. As a result, when the afterimage began to appear, those stripes looked red. The same thing happened to your rods, except since they only registered black and white, the afterimage was like looking at a negative of a photograph, dark replaced with light. That's how those black stars and stripes are white. So yes, human vision is fallible, but those mistakes that it makes can help us understand that wonderfully complex system. And that wonderfully complex system probably helped you learn about the anatomy and physiology of vision today, starting with the structure of the eye and its 3 layers, the fibrous, vascular, and inner layers. We spent most of our time exploring the inner layer, which consists of the retina and its 3 kinds of neurons, photoreceptors, bipolar cells, and ganglion neurons. And after learning how to tell our rods from our cones, we were dissecting how the weird flag illusion works. Special thanks to our headmaster,